Hello my dear student. Hope you are fine and safe at your home. Today I will take class about mitosis cell division. Today I will try to describe stages of mitosis cell division and importance of mitosis cell division. Hope you will enjoy this class. After watching this class, if you have any question about mitosis or equational, or you can say that constant cell division, please write down on comment box. I will try to give your answer as soon as possible. Welcome again. Well, my dear student, already you know that mitosis is a process of cell division takes place in the nucleus of a dividing cell, involves typically a series of steps resulting in the formation of two new daughter nuclei and finally dividing the cytoplasm of mother cell into two daughter cells which are identical to each other and also to the original mother cell in every respect. In mitotic cell division, the chromosomes of mother cell are duplicated and distributed equally to the two daughter cells. Hence, this cell division is called equational cell division. Starzberger and Schleicher in 1879 discovered mitotic cell division. German physician and cell biologist Walter Fleming in 1882 coined the term mitosis, and mitosis generally occurs in the somatic cell or body cell. So in mitosis we will get 5 stages. Mitosis is a continuous process. During this time the nucleus of the cell changes as rapid that is very difficult to explain it. Starzberger divided the mitotic cell division into 5 consecutive stages such as prophase, prometaphase, metaphase, anaphase and telophase. In each phase involves definite change within the nucleus. The main feature of different phases of mitosis now I am going to describe. First stage or phase name is prophase. At the early prophase, the nucleus become enlarged in size. Prophase is the first and longest phase of cell division before metaphase, during which the chromosomes become visible as paired chromatids and the nuclear envelope disappears. Definitable number of chromosomes appear by dehydration of chromatin fiber and removing of the nucleomembrane. Each chromosome has two arms joined together with a spherical body called a centromere. Except the point of centromere, each chromosome divides longitudinally into two identical halves called chromatids. In this phase, chromosomes become shorter and thicker due to more dehydration and phosphorylation of some protein by cyclin dependent kinase enzyme. By the end of prophase, the nuclear envelope and nucleolus disappear completely. The chromosomes remain scatteredly distributed in the nucleoplasm. By the end of prophase, introduce the creation of spindle fiber. So now I am going to describe about second stage, name is prometaphase. So this is the figure of prometaphase. Prometaphase is a short phase following prophase and preceding metaphase. The feature of these phases are the nuclear envelope and nucleolus break down and begin to disappear. In the phase, some proteinous bipolar fiber are formed which collaterally create spindle apparatus or aster ray. This is the aster ray. The place in between two poles of spindle fibers is called equatorial zone. Here the equatorial zone. All chromosomes of the cell get arranged on equatorial region or equatorial zone. Some fibers of spindle apparatus extended from one pole to another ones called continuous fibers. Some other fibers of the spindle apparatus remain attached to the chromosome of chromosomes are at equatorial zone called traction fiber or chromosomal fiber. Here this is a traction fiber or chromosomal fiber, but this is, you can say, spindle fiber. Each chromosome shows chromosomal dance when it attached to the traction fiber. In animal cell, spindle fibers are developed from the centriole, and in plant cell these structure developed from the microtubules. So next stage name is metaphase. So now I am going to describe about metaphase. Okay. In this phase, the main characteristics is metakinesis. Metakinesis take place, and at the end centromere divides. It is a short phase. This phase main feature are the nuclear envelope and nucleolus totally disappear. Each chromosome get horizontally arranged on equatorial zone of the cell with the help of traction fiber. The arranged chromosome appear on the equatorial zone as like as a plate. This plate in the cell is called equatorial plate or metaphase plate. The arrangement of chromosome on the equatorial plate is called metakinesis. The centromere of each chromosome on the plate is connected with both the poles by the traction fiber. With supercoiling method, maximum condensation occurs in chromosomes and they become very short and distinct. By the end of these chromosomes gets specific shape and the centromere starts to divide to form two daughter chromosomes. After that, most important phase name is anaphase. Now I am going to describe the main feature of anaphase. Here you are watching the figure of anaphase. In this stage, daughter chromosomes move towards the respective poles, and for this region it is called the anaphase or motion phase. It is also short stage. The main feature are the centromere divides completely, resulting each chromatid into daughter chromosome. So here you will get total number of chromosome 8, but in metaphase stage the total number of chromosome was 4, cause centromere divides completely, resulting each chromatids into 2 daughter chromosomes. The daughter chromosomes form the same mother chromosomes pulled away from the each other and start moving towards the opposite poles. In this movement, the centromeres are pulled towards, and the poles and the chromosomal arm trail behind. Most probably, this movement is caused by the shortening of the traction or chromosomal fibers. Due to positional variation of centromere in the daughter chromosomes, the chromosome appears V, L, I or J shape during the movement, and the name of V shape chromosome is metacentric, L shape name is submetacentric, J shape chromosome is called acrocentric, and I shape chromosome is called telocentric. So these four types of or four different shaped chromosome will get in anaphase stage. The anaphase ends when equal number of chromosome reached to each pole, and telophase begins. So now I am going to describe about the last stage of mitosis cell division, name is telophase. Okay, this is the last phase in mitosis, and you can say it is start when two sets of daughter chromosome reached in the opposite poles. The main features are, after reaching to the poles, chromosome completely stop their movement. A new nucleolus and nuclear envelope recognized or reorganized in each pole. The chromosomes again become long and thin due to rehydration, and finally form nuclear reticulum. These spindle fibers are also dissolved and disappeared gradually. A cell plate starts to form. A daughter nucleus is organized at each pole in the parent cell. Each daughter nucleus has the same number of chromosomes as that of the mother cell. The two daughter nuclei are identical in structure and characters. And after that, cytokinesis is the division of cytoplasm into two separate cells. The term first used by the Charles Otis Whitman in 1887 to explain the division of cytoplasm during the cell division. Cytokinesis usually occurs in telophase. Along with the formation of daughter nuclei after the nuclear division, this process of cytokinesis differs in plant and animal cell. In plant cells, cytokinesis is accomplished by the formation of cell plate at the equator of the dividing cell. The cell plate is formed deposition of small fragment, the phragmoplast or endoplasmic reticulum, small vesicular granules, lysosome like phragmosomes deposited at the equator to form plasmalemma or cell membrane. Hemicellulose, pectin, etc. are deposited to the thickening the cell plate. Finally, it joined to the cell wall of mother cell divides the cytoplasm, resulting to daughter cell. In animal cells, cytokinesis starts by the appearance of a shallow groove or furrow in the cytoplasm at the equatorial of the spindle. Slowly it deepens in constricts the cytoplasm, and the cell divides into two parts. In some cases, karyokinesis do not follow the cytokinesis, resulting multinucleated cell. These types of nuclear division is called as free nuclear division. Green coconut water liquid endosperm resulting from the free nuclear division. Multinucleated cell of algae and fungi are the called cynocytic cell. Multinucleated cell of slime molds are called plasmodium, and multinucleated cells of animal muscles are called the syncytium. So mitosis cell division is very much important to maintaining cell size and shape, growth and development also. It helps embryonic development and reproduction. Mitosis help in restoring wear and tear in body tissue and healing of wounds. Also, mitosis generates two daughter cells with the same types and number of chromosomes as the parent cell, and thus maintains chromosomal constancy of organism, maintaining the nuclear and cytoplasmic balance and produce red blood cell. Mitosis helps in cell generation of mitosis missing part to a different degree in multicellular organisms. On the other hand, uncontrolled mitotic cell division creates cancer. So that's all about mitosis cell division. Hope you enjoyed this class. After watching this class, if you have any question regarding mitosis cell division characteristics or stages, please write down or on comments box, YouTube comments box. I will try to give your answer as early as possible. Thank you so much for watching this class till end. Thanks again.